Yes, people on Zoom, please ask your questions too. Right. Just interrupt me.
So Farzan is getting ready to give you a formal introduction.
Okay.
Hello. Okay. So welcome. So thanks for coming to the talk of active matter seminar at CMSA. So today we're delighted to have Margaret Gardel from Chicago talking about active matter controlling field dynamics. So then, as they were saying, so like this is like in the Zoom, everybody feel free to just ask questions whenever. If you're on the Zoom version, you could either type the questions and we'll get to you, or you could just feel free to just speak up directly, whatever you prefer. And if you have any questions, comments, or concerns about the Zoom, or if you can't hear or see or whatever, just like message me if I was on that, and I'll try to.
Great. And Farzan, will you be monitoring the chat, or do I have to pay attention?
Yes, I'll be monitoring too.
Okay, great.
So you can just focus on the talk. Great.
And welcome Zoom audience. It's great to have you with us. And especially thanks to Margaret.
Yeah, thank you. That's good. So take it away. Yeah.
Okay, great. So thanks for having me. I had fun writing the abstract for this, although it was ambitious. So I ended up cutting one of the stories I wanted to tell. I am in the physics and the engineering schools here at University of Chicago, although I was a grad student at Harvard many years ago. My lab is broadly interested in understanding how macromolecules that are within animal cells self-organize into structures that allow cells to adhere to their external environment or to each other and or migrate and form multicellular tissue. And so these are two movies, not for my own group, but just because I think they're particularly nice. The left one is super resolution imaging of myosin 2 motors. These are these myosin molecular motors that translocate actin filaments in a cell that's migratory. And these filaments are assembling and disassembling and generating the contractile force that these fibroblasts generate as they move. And in the right,
let's start. Sorry, Margaret. Fibroblasts are wound healers. Is that their function normally?
Yeah, they're tissue remodeling cells. So they operate as individual cells. They bind to collagen and remodeling collagen matrix. So they remodel the interstitial space between like endothelial tissue and epithelial tissue. And then the right is an intestinal organoid. So it's stem cells that were induced to differentiate into epithelial, intestinal epithelial cells. And what you're seeing here is if you seed one of these cells and let it proliferate and divide, and you put it into a three-dimensional matrix, the cells will form a hollow sphere called an acinus. And the color here in both of these movies is different heights. So it's the same on the right hand, it's a membrane stain, and the yellow, the warmer colors are farther away from the cover slip. But you're seeing the shape of the epithelial cells in the tissue over time. So you can see a bunch of places of cell divisions, and you can see the shape of cells changes just in this organoid as it's increasing in volume due to cell proliferation.
So Margaret, the organoid is one cell thick, hollow shell?
It's one cell thick. Yeah, it's a single cell thick. That's called a simple epithelial tissue.
And it's growing through in a soft gel?
This is grown in a very soft gel, yes.
Right.
It grown in something like collagen or matrigel, and the cells secrete factors to disrupt the gel, and so they can grow in volume.
Wow. And has anybody tracked the lineages in the hollow shell on the right to see who begat whom and how?
Yeah, I mean, people have definitely done that in developmental processes. There's beautiful work done in mouse embryos. I imagine somebody's done that in these simple tissue culture conditions as well. I mean, especially with stem cells, there's really great questions of like, how do you, when you deal with stem cells, you would want to be able to study like microbial cell interactions, but in these cultures, the apical surface where cells usually interact with microbes is on the inside, so people can peel them open and put them on a soft gel, and so now you have access to that apical space. But yes, people definitely attracted. Boris Schraman has actually done some work on this in the 3D as of many others in the 3D structures.
Thank you.
Cool. Yeah, so the space, so I think about all these, you know, this motion is a reflection of the materials and machines that make up the cell. And for those, since this is an actin mod of, sorry, I don't need a lot of introduction here. The cytoskeleton and the actin cytoskeleton is the primary mechanism of this machinery. Actin filaments are semi-flexible polymers that catalyze chemical energy to generate polymerization forces of the polymers themselves. They're polar filaments. And then other molecular motors, these myosin 2, translocate actin filaments to generate contractile forces. And then other actin binding proteins sort of self-organize into complexes that allow forces to be transmitted either to the extracellular matrix, these focal adhesions, or cell-cell adhesions, okay? So I'm really interested, a lot of the molecular machinery has been now well characterized by tools of molecular cell biology, and with modern imaging, it's now you're able to track all these entities together, but we still don't understand a lot of the collective processes, all right? So I was really interested in contraction. So this picture sort of illustrates why I got really interested in contraction. And it doesn't illustrate the full thing, and I don't even know if I have it. I guess I could start with this. So, you know, the picture that people would think about contractility, the answer that you like to be given like 15 years ago or whatever, is that the motors are translocating actin filaments. And if you set up the motors towards the end, the pointed end of the actin filaments, and they move towards the barbed end, the other end, and if they're set up in this polarity organized fashion, you generate a contractile unit known as a sarcomere. And this is what's found in striated muscle. So there's this nice molecular understanding of the molecular motors. There is this one type of organization of actomyosin that people see in striated and cardiac muscle, which is this nice organized actomyosin. And I'm sorry, I don't have a picture of this. But again, like, you know, in the cytoskeleton and many disorganized systems, you see this random disordered collection of actin and myosin motors. And it's not clear in something with such high degree of disorder why a motor that is a translocation motor of actin filaments should ultimately lead to a contractile stress dipole in the actin network. Okay, so we spent a lot of time thinking about this, like how do you take these components of the actin polymers and this translocation motor of myosin to generate contractile tension?
So I'm sorry, another question, just the thing on the left, and maybe not your main topic today, but the myosins are at the bottom there. They're just all spontaneously self-assembled into this approximate rod-like cluster.
Yeah. Yeah, well, this is, so David, this is becoming one of my favorite questions. And I think I have a collaborator to help us do this now, because you need a real good biochemist. So this guy, Zev Bryan at Stanford, now can purify these motors in ways. It's an amazing question. Okay, so I will spend a few minutes. So yeah, so these myosin 2 filaments in non-muscle cells, in skeletal muscle and smooth muscle are all an assembly. They're consisted of a tail and then a motor domain that does all the activity, right? But this tail regulates the polymerization, okay? And in non-muscle and skeletal muscle, you have a very precise organization and of these, or size of these clusters. So for non-muscle, it's 30 motor heads per cluster, okay? It's always around that size. If you just take the tail portion and purify it, it's been known for some while that it polymerizes at sub-nanomolar concentrations of the protein. If you take the motor head on it, the motor head can be phosphorylated by a light chain on it. And if it's unphosphorylated, the head like folds back on it. And for some reason that we don't know, decreases the interaction potential between the tails and increases the like the critical concentration, we think, to something around 4 micromolar, so by 1,000-fold. And so then by phosphorylating that light chain on the head, it can unfold and then decrease the affinity. So there's this phosphorregulation in cells that regulates the assembly of these. And then not only that, but there's some higher organization of these, we call, of these filaments into something we call stacks that make them all register with each other. We think there's some weak potential interaction between just the tails that allow them to organize and interact with each other. So what we're seeing on the image, like that movie I showed you, now with super resolution, you can actually see where the tails and the heads are if you do the right fluorescent imaging. Each one of these bright spots here is not a single filament. It's a cluster of many of those filaments that have aggregated together. So it's sort of like it's a higher order oligomerization of these that we really don't know. We have no idea how that works.
Is there an understanding of the diameter of the myosin filaments?
So at a single filament level, that's been very nicely characterized by electron microscopy.
But why? So it's been characterized, but why? Well, how does it know to have that diameter?
I think, yeah, so why do you get to a filament size of 30? I mean, yes, we don't really have a good answer.
All right, thanks.
And I mean, I think and also so smooth muscle forms these nice side polar filaments. I mean, smooth muscle is such a cool motor filament because the polymer, the myosin polymer, can change from being like a few heads in size, like to up to several thousand. It can actually change. It's much less constrained and regulated. And then, so it's called a side polar filament, and the skeletal is very large. It's like 300 heads or something like that. And then the non-muscle is 330, but there just hasn't been a lot of biochemistry and sort of macromolecular scale biophysics done. I mean, it's a great, there are some really great questions there that really are at the heart of how you control force generation, right? I mean, I mean, this is the motor that's actually generating the active stresses, and we really just don't understand how it's controlled.
Yep.
Okay, cool. That was probably too much time for me to spend on that, but I think it's great. Okay, so yeah, we focus mostly at this scale. We try to skip everything. We try to hope that everything at this molecular scale is understood, but as I just explained, that sometimes it's not, and there's some interesting questions there. But we focus on these larger scales, and we spend a lot of time thinking about how, some, self-assembly and force transmission questions at the molecular sort of like to larger scale phenomenon. But then also we've been, which I won't have time today, is like ways of using more continuum or coarse graining approaches to describe the mechanics of cells and multicellular tissue. And I'm not going to say anything more about it, but in the past 2 years, we've been taking some of our old data and our old studies and now working with Vincenzo Vitelli's group here to develop machine learning approaches to then do physics guided machine learning to really learn the physics models of these cells. And by taking information from the fluorescence data. And I personally think this is the way we should be doing. I've been completely sold by this approach for anything that's any kind of active matter, basically.
So machine learning is going to tell you some model that is the only possible model for this? Or?
Well, so this is a controversial topic, right? So we've done it a few ways now.
Right. So I'll retire as a theorist.
Yeah, no. So what we did first, so we published this, is we used, there's really nice hydrodynamic models of active pneumatics. Okay. So the first thing we did was show that the machine learning could learn the parameters that we put in a hydrodynamic model for active pneumatics. So that's one way you could use machine learning. It's just as a quicker, right? You're not learning anything new there, right?
That's like glorified curve fitting, but
exactly, it's glorified curve fitting, but it's a lot faster than what you would do if you had to go through simulations. Okay. Okay. All right, and then so here, what we're doing with especially data like this. So in this movie, we're taking images of like what the actin cytoskeleton looks like and making measurements of force measurements. So many years ago, we had actually worked with Christina Marchetti's group, who had built models of active gels as a picture of making predictions of the force transmission to the substrate on adherent cells. And the model worked pretty well, right? I mean, it worked pretty well. And so what we can do is then say, can we get anything more information from the data and from the images by that we weren't able to pick out parameters with our eyes, right? Our eyes are like, we have some intuition, but we were just sort of going through and picking out what things we thought were important. And what AI is doing for us in this case is telling us which features of the images are important or not to make an accurate prediction. And then once we can make an accurate prediction, then we take into this trained neural network and say, what features of the image is it pulling out? And it's giving us length scales that we think that are important. And some of these length scales are continuum length scales, but some of these are very local. And I think this is one of the key challenges of active matter is that you have this coupling between these very disparate length scales. Like you have this biochemistry going on, but then you have this continuum mechanics going on. And you never quite know where to coarse grain the system. And so what we're seeing by doing this, like, you know, attention network mapping on the trained neural network is a way to pick out the critical length scales in the problem. And so for the case of the cells, it picks out length scales on the scale of those focal adhesions, the self-organization of where those adhesion complexes are forming, but also the continuum length scale of what the mechanics of the network are. So that's the part where I think it's doing a job. It's still not telling us what the physics is, right? And so what we're just doing now, and this is like 2 weeks old at this point, is now taking the continuum model that we did with Christina Marchetti and Sheila Banerjee and saying, like, now, does a machine learning do anything? When we put some parts of that model in there, we have more confidence in some aspects of that model, and there are some places where the model didn't work. So now if we take that continuum picture where we think that it's working pretty well, but now add that ability to optimize the model based on the features of the focal adhesion sites, can we understand how to do the model better? And is that model the most efficient way to describe the problem, right? That's sort of what, so there's like this combination of both like theory and sort of making predictions that I think is very helpful. It's not like the machine is telling us what to do, but it's this rapid iterative feedback between like me, the experimentalist, Vincenzo, and the machine that helps us get to answers much quicker than we had in the past, which I think is super exciting. No,
it sounds fascinating. I look forward to see what comes out.
We'll see. Maybe we haven't published anything yet. So
I mean, the standard worry would be that maybe the most efficient model isn't what nature has evolved for things as complicated as cells.
Well, I mean, this question is, what is our goal, right?
Right. Sure.
Yeah, what is our goal with modeling?
Right. So just to make sure I understand, so does the machine learn, does it tell us actually what terms we should even include, like terms that we haven't thought about, or is it just, so we give it the terms and it just tells us the relative coefficients?
That's right. So we've done it both ways right now, but like, you're right, the limit will be what we can predict and how useful. I mean, I would argue that we want models for is also to be able to engineer the system, right? We want to be able to make a perturbation that was something we know we can do, right? And then have the cell do something different, right? But there's a lot more work needed to be done to see if it makes it. I would argue that all of quantitative cell biology and all our attempts to make mechanics models have been, when they work in a very limited scope. It's very hard to make predictions, right? And hard to do engineering at the mechanical level. And the hope is we could expand that, right? Okay. Okay. So I spent a lot of time talking. Okay, so we work at these two different scales of the sort of at the microscopic scale, but then also this is the work that inspired the initial collaboration with Vincenzo, the continuum modeling work with Christina. So I'm not going to talk about that today, but we think about these two different scales. I'm going to summarize work we've done about the microscopic scale. This is to sort of summarize what we think of as the phase space of active materials that are made out of semi-flexible biopolymers and motors. I'm going to start from there. I'm going to summarize a bunch of work. And then I'm going to move the last part. I'm just going to talk about work that we were doing in cells. And the key thing with cells that I think is different than in vitro is that the parameters are adapting and changing over time, and how we can use optogenetic approaches to study that. Okay. Okay, so for active matter that's constructed out of actin filaments and myosin, I first want to just give you what the phase space of actin networks are. Okay, so if you think about like what solutions of actin, even without any cross-linkers or motors, there's this whole range from semi-dilute solutions of just polymers that are sterically entangled. And then if you crowd them, there's an isotropic to pneumatic phase transition at very high concentrations that allows them to form a liquid crystal. So this has been described for a long time. This liquid crystal phase requires the actin polymers to be quite short, so the relaxation time stay reasonable with a liquid crystal. So if you take longer filaments and push them toward that isotropic pneumatic phase transition, you get a lot of long filaments that are just have sort of traps, right, are kinetically stuck and can't relax to form a nice pneumatic phase. Okay, so then if you add in cross-linkers, so proteins that provide cross-linking between filaments, you now can have a phase space that's two-dimensional. And I'm just going to make this simple for one type of cross-link. And the two important things we think about this cross-linker are, one is it comes on and off the actin filament, the biopolymer, but also this cross-link is highly flexible. So it doesn't provide a lot of mechanical constraints to the orientation of the two filaments it's cross-linking. Okay, so it's like two dynamic binding sites with a long flexible polymer is what I'd say. So now we have at a single concentration and at a semi-dilute concentration, for as a filament length we get longer, at a single cross-link concentration, you would have at the long filaments, you get these nice viscoelastic networks. These are gels that assemble, that are kinetically trapped gels. You can go through different mechanical regimes of the gel where energy is stored either in filament stretching or bending as you apply shear stress to the network. The surprise to us a few years ago is when you form actin filaments with these long filament dynamic cross-linkers, below this transition of we thought before this would be like a gas or a cluster and a network phase, there ends up being a liquid phase, sort of like a pneumatic phase, where this cross-linker is inducing a weak transient attractive interaction, and you get pneumatic-like isolated tactoidal droplets of the cross-linked actin in solution. So if I played a movie of this, these little footballs would be diffusing around in space. So they're not stuck. So they're little.
Margaret, another question, please. Is there information in the angle that controls the pointiness of these footballs?
Yeah,
tell you something about
so you can change we had a paper on that, I guess, a year ago, and you can change, or maybe that was actually in Tim's original PNAS paper. You can change the shape of the tactoid by changing the length of the actin filament, but also the concentration of cross-linkers. So you can change the isotropic and anisotropic surface tensions that way.
Okay, and then another question: how monodispersed are the actin filaments?
Oh, they're really not. I know this is one of my surprises. There's a very broad distribution of the actin filaments when we actually make these in practice. I know it's a little bit surprising you get something that's such a nice pneumatic phase.
I see. What controls the length of the filaments?
Okay, so we have to add, we need to add an additional protein called a capping protein.
I see.
Upon the polymerization, and then the stoichiometry of that capping protein controls the actin filament length distribution. There's a broad distribution, so when we add sufficient capping protein, the longest filaments should really be no more than 5 micrometers. So maybe we're really just suppressing it down.
Okay, and then thank you for being so patient with me. What determines the spacing of the tactoids in the picture in the upper right? Are they repelling each other or somehow?
No. And actually, so if I had a movie of it, they merge together and they anneal.
Oh, okay.
So the spacing here has something to do with the time at which we prepared the sample and the initial concentrations. Yeah.
Okay. So they nucleate. It's a first order transition.
It is. It's pretty. Nucleate. I know. Okay. Yeah. It's very cool. And then just so you know, like if you add myosin motors to this, I don't have time to talk about it. The myosin motors actually go to the center of the tactoid because you can imagine the liquid crystalline order in the droplet. They move passively, we think. We don't think that's an active process. But then there's something about the motors that allows them to interact with each other to form this like aster that then makes the droplets divide in two. Yeah, it's so fun.
Good. And what was going on? By the way, we don't have a hard deadline at 2, so don't worry about that. But the picture, the movie in the lower right, which had these dancing filaments that were coming together.
Those are threads. Those are liquid threads.
Those are actin threads?
Yeah, so what those are, so this is a movie, there's an arrow right here that goes right at this transition. So a viscoelastic, I don't have a movie of the viscoelastic network because it would just be stuck. It wouldn't be doing anything interesting. These are threads, liquid threads of cross-linked actin that are, and it's a movie is over time. So the thread is formed because you add in a bunch of cross-linker and the actin filaments aggregate together, but there's a slow relaxation time. So what you're seeing is surface tension driving the contraction of these liquid threads, just like a liquid of polymer fluid.
So, okay, and so those are like elongated tactoids? Can I think of them that way?
And over time, you could see some of these start to have tactoidal features, correct? Okay. And so if you waited a long amount of time, if the filaments are too long, they would become tactoids, correct?
And only a subset of the filaments are fluorescently labeled, or everything's labeled?
Everything is labeled here.
Thank you.
Yeah. So this, so in the semi-dilute regime, and this is what I think is really interesting here. So I've not appreciated that there would be a liquid phase of cross-linked polymer networks. This was a complete surprise to me. And as Dave Waits would make fun of me for, I've been studying actin filaments since, the 90s, and Dave Nelson would tell me too, like I haven't changed my field like I'm supposed to.
That's okay, you're doing fine.
Anyway, so this was a complete shock to me that you could form a liquid phase. So as far as so Dave, Dan Needleman, right, one of your colleagues, had this hypothesis that the mitotic spindle behaved like a liquid droplet, right? But until this point, now we know the mitotic spindle is a cross-linked network of microtubules. But I think this was the first time we could see something experimentally where we knew the components that we'd actually form something that's like a liquid phase. I mean, so I think you don't really need a lot of activity to form a liquid phase, is what I'm saying, right? This is a passive system.
It's kind of an actin homologue of
right.
What Dan is talking about.
Exactly, with much fewer components, right?
Yeah, sure. No,
yeah, so and I just want to also, I play this also too, because like surface tension does a good and fancy, I mean, people in actin matter love things that change shape, but we forget about surface tension sometimes. Anyways, okay, so then if you go down really low, you get a gas. Okay, all right, so now I want you to remember this as we add motors to this. So now we're going to add molecular motors, myosin 2 motors to these systems, but you have to keep this in your mind. And most of the time, I'm going to be changing the filament length and then keeping the cross-link concentration really low, so that as I change the filament length, you can go between a network, a gel, and a liquid phase, okay? Sometimes when we're in the gel phase, we go up and down this vertical axis to change the cross-link concentration, and and sometimes we change the type of cross-linker, okay?
And sorry, so another question, is there a sharp transition between stress strengthening and stress weakening? Strength, in the A and the B on the right, is that a sharp boundary?
It's a pretty sharp boundary. I mean, that was what I did my thesis on, David.
Okay. I should remember. I'll probably find it.
So yeah, no, I mean it's a pretty sharp transition. It's that it's like a phase space as a function of the cross link of but the stress weakening regime is less well defined than the stress stiffening regime. Yeah.
Okay.
But I mean, in terms of what kind of phase transition is, it's not a phase transition, right? It's not a first order phase transition.
Okay.
Yeah.
Thank you.
You or somebody else, I think, told us that we couldn't call it a phase transition. I remember this quite clearly.
Yeah, as usual, you ignored me.
No, I don't.
No, good. Go ahead.
Okay. All right. So now if we're working with short filaments that would form a pneumatic, when you add motors to it, Zvonimir Dojic has showed this really nicely for the microtubule system. We saw this for the actin system about the same time that we discovered this the pneumatic phase, right? Like, so a cross-linked pneumatic phase. So, this is the actin, homologue of the, whatever it is, of the Dojic system with microtubules and kinesin. Okay. And I'll just put a plug in for the actin pneumatics for a second. Because I can, there's a few really cool things. One is you see that we see the myosin motors, they're really gross looking. And this has something to do actually with David's question before. I really think it has to do with this oligomerization state of the tails, allowing them to build these high order structures that we don't understand. But the important thing is,
so the yellow are aggregated
motors. Yeah. Yeah. And I mean, I guess this, and as one of my theory colleagues said, this is really telling you the power of hydrodynamics, that you get something that really looks like an actin nematic, even in light of all this heterogeneity in motor density and location, right?
Mm-hmm.
I mean, it's really amazing. I never thought you would see something, because when I show you the gels, you know, oh, I'll show you the gels, actually, it's right here. This is what the gels look like. It's just a bunch of crap collecting together. It contracts, right?
So the gel thing on the right, there's no hint of pneumatic order? If you
No, well, there is at the beginning, but the motors quickly disrupt it, right?
I see. Okay.
And they quickly disrupt the pneumatic order. And this is probably one of the more pneumatic looking gels. Yeah, so the motors quickly destroy the pneumatic order that's formed as you, because all these samples are made by actin filaments crowding on a surface. So you do get some of this pneumatic order when you crowd the actin filaments that way. Okay. Okay. So, so what do I want to say about the other thing about the actin pneumatics as opposed to the microtubules is we really think is a like a liquid crystal, not a liquid crystal elastomer. I mean, so that if you look at if I look at all the liquid crystals and pneumatics from the microtubules, they're all made out of these bundles that are sort of acting like one-dimensional extensile bundles. This is really acting like a pneumatic liquid crystal. So it behaves, and we've done a bunch of studies with these hydrodynamic simulations that it works, all the theory works very well for these systems. And we can change the splay and the bend elasticity of the system by changing the actin filament length, right? So changing the filament length can change the elastic constants of the liquid crystal, but also we can do things like dope in a little bit of microtubules, and that really increases the bend elasticity to really change the elastic properties of the pneumatic liquid crystal quite a bit.
So, so the kind of self-stirring system in the middle, that's between two plates that are with a gap that's narrow compared to
so this system is actually experimentally, it's exactly what was developed by the Dojic, by Zvonimir. And it ends up being so when we first started working with the gels, and I think we're going to go back to it pretty soon, we were working on 2D systems crowded down to the surface of a passivated glass cover slip and or lipid bilayer, basically to make the system as unsticky as possible. Really, when you're dealing with gels, if you want to model like an adherent cell, you would really love a system where you can control where you're grabbing onto things so you can build tension, because I think I really want to understand how high different amounts of tension and force within the actin-myosin allows for different biochemistry to occur. And in the pneumatic, the forces are always very low. Go
ahead. Sorry.
Yeah. So sorry, I didn't explain the Dojic setup. So the Dojic setup is now, instead of that, you put an oil interface on top of a cover slip. You have a surfactant on top of that oil, and then you crowd the actin filaments or the microtubules on top of that onto the surfactant, and then you put you have aqueous buffer above that, and it's exposed, so you can add whatever you want on the top while you're imaging, and you're imaging it from below. So it's a really low friction boundary condition.
Okay, and regarded as a fluid mechanical system, it's thin, and so momentum is bleeding out of the system all the time. That's
right.
It's not conserved.
That's
momentum
right.
Here.
That's
and
right.
Then the take-home message from these two pictures on the right is that extensile fluids tend to self-produce pneumatic order. Contractile things like gels don't.
Oh, self-renew. Yeah, this keeps its pneumatic order. The extensile fluid keeps its pneumatic order. I mean, let me go to the next slide. So this is, if I summarize a bunch of work, we spent most of our time focusing in the gel phase. And it turns out in the gel phase, the reason actin-myosin contracts is because of the asymmetric and nonlinearity of actin filaments, right? So when you apply, it's Euler buckling, right? When you apply a compression stress, the actin filaments buckle out of the way. When you apply tensile stress, they can withstand a lot of tensile force. So there's this whole regime where the filaments are soft, like actin filaments, and there's enough connectivity to build up compressive and extensile force that buckling just relieves any of this compressive or extensile stress, and you're left with contraction. And then it turns out that if you tune the filament stiffness high so that you exceed your motor force can't exceed the Euler buckling force, now you can still get contraction. And Dan Needleman's group has described this one as well, where if you have dynamic crosslinks in the system, those crosslinks can accumulate when there's a maximum amount of overlap of your filaments. So what happens is as you start to increase the overlap of your polymers, there's an accumulation of crosslinks, and that basically acts like a high friction, so you can't get extensile force. Okay, so this is in the contraction phase. The cool thing is as you decrease your connectivity, so you go left, whether you do that by cross-linking or by filament length, there's a regime when you decrease your cross-linking that basically you change this contractile dipole to one that's net extensile. And Zvonimir described this years ago as that you have a higher likelihood of binding when your filaments have higher amounts of overlap. So you end up with probability distribution of something that's net extensile. Okay, but this phase space sort of summarizes why you can take a translocation motor of two filaments. And obviously we're only assuming the anti-parallel organizations. When they're parallel, nothing interesting happens. You can change it from being extensile to contractile dipole, and the shape of that contractile dipole can change depending on the mechanics of the system. Okay. And then overlapping this is the importance of the liquid to gel, right? They're not exactly, I don't exactly know if the boundaries are the same. We never went that far, but this extensile regime happens to fall into the regime where you get pneumatics because it's short filaments that are, not well connected. And the contraction happens to fall into the regime where you get gels. Okay. All right. So this is all I wanted to say. This is like my brief summary down, which is not too brief. Down, like sort of exploring different regimes of active matter that I know based on what Farzan had told me before, you heard a lot of different talks. I think I was super excited when there had been a lot of observations in the field and we could bring together all of these to understand transitions between these different phases. But if there's any questions on this before I go, and I promise I won't keep you too long. Okay. All right. All right, so the next part that I want to talk about is I guess in the past couple of years, I've become more interested in going back to thinking about those first movies of cells. I think we have a pretty good handle on how to describe the activity, right? Like now that we have that sort of phase diagram, I think active stresses, we sort of have an idea of how to go about doing that. The problem I see with going back to cell morphogenesis is that the parameters, any of these active or passive mechanical parameters, are really changing over time. And we haven't had good ways of knowing what causes those transitions or how to make predictions about how those transitions are occurring. So, and this also happens in the active gels, right? There's a lot of structural changes that are also changing the mechanics.
So Margaret, in the cell case, the changes in time that you envision are due because of different gene expression patterns? Or
I think they happen before that. I think they just happen because the biomolecules, have force activated chemistry. So let me give you an example. So like sure. In those stress fibers that I showed you, those contractile bundles that are spanning the cell, if you pull on them above a critical force, they'll start extending infinitely, right? They're infinitely extensible. And the reason is because there's molecules in there that sense extensile stress and then mediate assembly of actin polymers and myosin motors in places of mechanical failure. So you can, in an infinite reservoir, you could just pull that cell as much as you wanted, and it would just extend at a certain yield stress, right? And, so and it would never fail.
Okay. So when that yield stress is exceeded, that you bring in extra material to keep it from thinning out too much?
That's right. And likewise, in the opposite direction, we know that there are other biochemistry happening under compression.
Right. And then this set of parameters that you've used to illustrate things, eta is a viscosity.
It's an active stress. Yeah.
Okay. So an active stress.
And a viscosity and an elastic constant. I mean, it's the simplest thing you can do.
Good. And sigma in your mind is a surface tension, or what is sigma?
Yeah, this was a bulk. It would be a bulk stress in this case. It's a bulk, but yeah, in our actual model.
Spring constant. Okay.
Yeah, I mean it's not supposed to be contained. It was supposed to be illustrative of this.
Yeah. Okay.
I mean, the other problem when we do any kind of modeling of, say, epithelia that I ran into when we started doing this is that my feeling as an experimentalist is that all these parameters are spatially varying. And we also know this in cells, like that there's different parameters. And so it just, the number of parameters we have just, you know, gets out of control for anybody who wants to do this in any kind of rational way. So how do you deal with this problem?
Machine learning, of course.
What?
Machine learning.
Well, you still need experiments, right? But yeah, machine learning can help a little bit.
Even better.
Okay, so this is sort of the way I was thinking about it. Like, so if you think, so I've been thinking about this complement, and this is really inspired from conversing with my colleague, Arvind Murugan, who really thinks about adaptive materials that change over time. Is like, you know, I had been focusing on this problem of active matter, right down here, of this sort of feedback between force transmission and force generation. But then if you want things to adapt, you also need some force sensing that then feeds into this, right? And these coupled together allows living matter to do what it needs to do, right? And so this is sort of the challenge that we've been working on. And in the past 6 years ago, we've been using optogenic approaches to do this. And so I just want to give you a flavor of this. Okay, so the what controls contractility in cells is a signaling molecule called Rho GTPase, which gets turned on and off depending on different cues. It's mostly located near the cell membrane, and different Rho GTPases. Well, Rho A controls the assembly of actin polymers and myosin 2 motors. So when you turn it on, you get a bunch of assembly of actin and myosin. And with different Rho GTPases, you get different kinds of actin-myosin structures. Okay, so if you can, with biosensors, you can visualize where Rho is getting turned on and off in the cell. And so this is now like a chemomechanical feedback, right? That's happening, right? You have the Rho turning on and off at different times, and that's changing the mechanics. And it becomes, and so what had been observed in sort of development and then also tissue culture when you use these Rho A biosensors is you see dynamic signatures of Rho getting turned on and off in a spatially controlled way, okay? And so people have been able to characterize that Rho, these pulses of Rho is where actin myosin gets turned on. This is where contraction happens. But the problem is that the way you could control this is by like knocking out a gene that then controls one of these rows, and then you get something totally disrupted. So, and it led to this really cool idea of like the cell cortex being an excitable medium where you have this mechanochemical feedbacks happening. Okay, so so the optogenic approach that we can use is now you can use light to control where Rho A or any signaling factor that you want is turned on and off as a way to probe these dynamic adaptive and active materials. And this one we use is based on this light sensitive change in conformation of this LUB2 J alpha helix protein. In the presence of blue light, this J alpha helix unfolds and exposes and has a high affinity interaction with this other PDZ protein. So what we do is we target half of this component to the membrane and the other one to this Rho GEF. And so in the presence of blue light, they come together with a high affinity and it's reversible. So this is taking a micro mirror array and blue light and shining it on a portion of a cell, and that's the localization of this Rho GEF. And you can see that it accumulates very rapidly and goes away very quickly as well.
So the size of the fibroblast, sorry, Margaret, the size is about 100 microns or what?
Yeah, maybe 30 micrometers.
30.
Yeah, and
okay.
I mean, I will say, yeah. So this was one of the first, our colleague, Michael Glotzer, had been one of the first ones to develop this. There's now a lot of optogenetic tools. They're still really hard to work with. I think of the next couple of years, they're gonna get easier. But it turned out that it was pretty unique for this one that we got this subcellular confinement, that a lot of them work that are very diffusible in the cell. And so you wouldn't be able to get this subcellular resolution.
So just make sure I understand how the optogenetics works in this context. You're using light to activate transcription factors?
No, we're just using light to spatially concentrate a protein that then biochemically turns on row A, and then leads to the accumulation of leads to polymerization of actin and polymerization of myosin.
Okay, so
you could do this with transcription turned off. There's no it's just a bag of proteins right now.
Okay, and then the light is typically turning on every point in the cell, or it's important that you can control spatially within the cell where you turn it on.
We are still learning how to use it. We like having spatial control over this for the subcellular stuff. In tissue scale studies we're doing, I think you could go to a whole cell, but it has been nice to be able to do subcellular, because then there's differences in different regions.
Okay, and then you get a different response when you shine it on the nucleus as opposed to the cytoplasm and stuff?
This one, the nucleus.
Okay, fine, thank you.
Well, the nucleus, I'm not sure. I mean, I don't know how to ask you. Yeah, like there's, we've shined it in lots of different places. Most of the time we don't see a response on structures. Well, all the time we don't see a response on structures that we don't think are affected directly by Rho. This is a short time experiment. This is like in the course of a minute or two, right? And then we're seeing a response very quickly, and that's sometimes reversible and sometimes not. So that's right, yeah.
Okay, so the right-hand side is a confluent cell
that's right.
matrix.
Yeah.
And the left-hand side is what?
Is a single fibroblast, and we're just shining it on a region within the stress fibers.
All right, at an angle. Okay, thank you.
Yeah. So to make I'm going to focus on this epithelial tissue just for 5 minutes. This was a more recent study where we shined it on a junction, and so this junction is an actin-myosin belt here. Sort of as you can see from this movie, for this 5-minute pulse of light, the junction shortens. We just measure the length of the junction. It shortens. You turn off the light, the junction goes back to its original length. So this is something that's highly reversible. And this would be consistent with any of these vertex models that people use to develop epithelial tissue. But now with light, we can control the magnitude and we can control the duration of which we're doing this. And so if we just double the length of the pulse we give it, it becomes irreversible. So, we increase the length of the pulse, and now we see this irreversible shortening of the junction, meaning that we've done something, the cell has done something in response to this signal that now makes this an irreversible shape change, okay? Yeah. Okay,
so the working hypothesis, there's some sort of polymerization that's going on there?
So just for sake of time, so what we think is happening is that the force is, there's an increased tension that's happening that's initiating endocytosis of adhesion receptors and remodeling the junction. And that process has a kinetic rate associated with it. And so for short times that doesn't happen, and longer times it does. But there's like a compression-induced endocytosis of adhesion receptors.
Thank you.
Yeah. And if we like inhibit the endocytosis, then you don't see this permanent length change. But you know, with optogenetics, you can look at this transition between something that's reversible to irreversible. And so now you can ask about this adaptive nature of how things are remodeling over time. So we can go back to like a vertex model, and there are lots of proposed mechanisms of adaptation and changes in tension. So our theory collaborator, Sheila Ditcha-Banerjee, who's at Carnegie Mellon now, and their student, Mike Stadden, you know, checked through these. And the only model that worked with our data was one they cooked up was basically a strain thresholded remodeling of the junctional tension. And so that if the junction ever gets contracted to a certain amount compared to its rest length, that sort of initiated some change in the tension at a certain characteristic rate. And so then the cool thing is now with these dynamic models with optogenetics, we can test it with the experimental data. And so we can go through and change the different durations of light and compare it with the model. And then also change the amplitude to see how they are consistent with the model. So this is a way of testing these dynamic models with this optogenetic stimulation of varying the profile of the stimulation. Okay.
So the models that you showed in the previous slide, if I remember well, if you could go back one slide.
Yep.
Yeah, so that's a, it's sort of, the red line is anti-symmetric. I mean, what, does it have to be?
Yeah, it's anti-symmetric.
The strains have to be that symmetric? It seems strange.
It doesn't need to be. This was the simplest model they could cook up that allowed it.
Okay.
Yeah, we tried to minimize the additional things that were in there. So yeah, there's this constant rest length remodeling, but then there's this strain folder remodeling depending on whether the system is under compression or tension. It either extends or it compresses or shortens.
Thank you.
How do you distinguish between
Andreas?
I needed to ask a question, but yeah. I don't know how long the talk takes. I'm here at Harvard. I'm visiting here.
Oh, wow.
So for a year, actually. Now, how do you distinguish between the single cell and the tissue response then? I mean, you do a local
there's
for one cell.
Yeah, so there's no changes in length of the neighboring junctions. And also, I guess the other,
sorry for interrupting, but it would, from the vertex models, it should kind of, because
I know, I don't believe in any of the vertex models anymore. I'll just let you know that.
Okay, good.
No,
good.
It's totally wrong. Good. Yeah,
okay,
it's
good.
Totally wrong.
Okay, good. Good to know. Good to know. Thanks. Now I know. I didn't know.
No.
But that would prove that they are wrong, right? That would prove that they're wrong, what you just said, right? I mean, if you just...
we have lots of different data that says that they're wrong.
Okay, got you. Okay, interesting.
Sorry to say something controversial at the end, but yeah, I agree with you. There's nothing consistent here with the vertex model. I agree.
Got you. Oh, but that's very important insight, I guess.
Yeah. I feel guilty because Lisa Manning and I published a paper using the vertex model, so I should be careful. But yeah,
that's why I trusted it. So that's why.
I'm sorry. No, it's really a problem. We had to fight for a while. Okay. So, but this is where like what I think of as a cool tool, besides being able to change the structure, is now you can take the same pulse of light, the same amplitude and duration, and just break it up and to really dissect out these different kinetic processes. I should say that Kate, who did all this work, is a developmental biologist. So she really wanted to make the full junctions remodel like you get in development. And she was bothered by this saturation that happened. And what the model predicts is that if you do break up a very long pulse into two shorter pulses that sort of allow for this remodeling, you should be able to overcome this saturation in the contraction. And so you basically get through two of these acute periods of tension remodeling to allow you to get to a shorter state. And that's what's predicted in the theory, and that's what she's able to see in the experiments. And what I think is cool and sort of like a design process here is like, living systems constantly are subjected to lots of force fluctuations of pretty high amplitude, right? You'd want them to be sort of robust to these. And so this is telling you a way in which they might not take every large amplitude force fluctuation and dramatically change your shape. You'd have some threshold here that would need to occur over a certain amount of time that would sort of dampen those fluctuations to allow you to sort of do something in a more, calm fashion. But this is also, I think, a way that optogenetics can really help you dissect out all these different kinetic processes. Okay. All right. All right. This I already answered because I'm not going to say much more than what I already did when I answered David's question. Is we can visualize what's happening in the membrane when the contraction is happening. And it looks like there's endocytosis of adhesion receptors. This has been described at junctions as the way that they remodel. And if we treat with a dynamin, an endocytosis inhibitor, you basically don't get any permanent shortening of the junction. Okay, so there's a lot more sort of, I would say, mechanistic cell biology to understand how this process is happening. But this was our idea of how do you build a ratchet that allows you to shorten your junction length in response to fluctuations in contractile stress.
Margaret, is your idea that the endocytosis goes into both of the cells that that are connected?
Yeah, that's a good question. Yeah, we haven't done enough studies to know like if it's how it's partitioned. I would assume that whatever membrane components, yeah, you could imagine happening in both into one cell or separating off into individuals. We don't know.
Good.
Yeah, we don't know. Okay. Yeah, so I don't know, like I just wanted to end on this. Like I've gone through a period of my midlife crisis thinking that we had sort of like, I didn't know what we were gonna do. I shouldn't admit this at the end of a seminar. but like,
I can't believe you. That's a lie. Make it up.
But like the optogenetics is a really exciting tool because it's something that we've always wanted to be able to do. And if I think about all these different active materials that you can find in different systems, I think it's making these questions really amenable to experimentalists, right? To be able to ask really difficult questions. And I guess want to sell that approach to people who think about mechanics, because lots of people in optogenetics don't care about mechanics at all. But for people that do care about active matter, I think it's a really great tool. And if you want, they're hard experiments to set up. We're starting to do some of this stuff with motors, again, with Zev Bryan at Stanford, in vitro. They're great ways to set up questions about active matter. So I think the field, sort of experimental field, needs to work together to make them easy enough for lots of people to do. That's sort of what I wanted to end on. Cool. And the people whose work I talked about, there's a bunch of, the early stuff is really a collective effort over the course of 7 years from many previous postdocs and students. Kate Kavanaugh, who had just graduated my lab, is now a postdoc at Caltech. But thank you.
Thank you.
Thank you for the great talk. We have a lot of questions. So are there any more questions from?
Can I just have one more quick question, like about your experiment using the optogenetics?
Mm-hmm.
So when we kind of want to contract those edge lengths, could you comment on what determine the time scale that the edge lengths shrink, and like after you release force, it also kind of relaxes.
Yeah.
A little bit, yeah. Like, are the two time scales the same, and what determines the time scale?
Yeah, that's something we really ignored that. Yeah, that's a good question. We ignored that for a few reasons. Yeah, so this, we assumed that this has to do with some viscosity, effective viscosity that is reacting to the force, but we actually don't know whether it has to do with some kinetics of accumulation of actin-myosin on the junction, right? So like when you shine the light, there's a kinetic process of myosin accumulation, right? That could tell you a time scale of which force is built up. So in our simulations, we approximated it as a step function, which is experimentally not necessarily true. And there also could be some resistance, some drag that's happening in the system, but where exactly that drag and resistance is happening, it's not clear to me, if I'm being completely honest. And the other thing that we sort of swept under the rug here is that, so all these systems of length changes people usually study epithelial tissue in developmental systems. We use this this epithelial monolayer that's built out of KCO2, human intestinal cells, which are very stationary, but they're very slow to respond to Rho stimulation. So that also could have been a reason. So I don't know, there could have been like the total amount of adhesion stress. That's a great question.
But should we expect them to be identical for every edge, or they should vary from edge to edge?
Oh, this was one of the weirdest things about these experiments. Is like, so I show you this data, Kate performed these experiments over the course of 9 months, with many different junctions. This behavior you see, as long as you normalize for the initial junction, the data is very reproducible, and as Andreas was saying, like all these things that you'd expect to happen in terms of force balances from neighboring junctions or the angle, you'd expect all these to affect how much you would contract, and none of those were important at all. It was very consistent.
Even for the data on the next slide, which we impose the light for a longer period of time.
Yeah, all of it. Yes, all of it. Yes, exactly. Yes. Yeah, it was way more reproducible than it should have been.
Great, thank you.
That's fascinating. So can you just quickly say what the system really is? Is that now on a glass surface, or is that?
Yeah. So Andres, this is a really this is actually becoming. We're realizing this is really important. And I didn't get a chance to talk about this, but these are epithelial tissues that are formed on top of a collagen gel, like a real physical collagen gel. And so there's no focal adhesions. And what we're realizing now is that all the active stresses in this epithelial tissue are dominated by cell cycle stresses. There's no, but then if you suppress the cell cycle, There's no motion at all. It's like CDK1 dependent. It's like, you know, it's this other, and then if you put cells where this whole system is suppressed, but on glass, where they can form lots of nice focal adhesions, they move very nicely. So they're migratory. So I actually think in epithelial tissue, there's like these two, and this is all stuff that we're trying to figure out. There's these two sources of active stress. There's like the cell matrix interactions that have to do with motility. And then there's this cell-cell interactions that have to do with the cell cycle that are really a source of noise in the system. They don't actually, they just make the cells sort of jiggle around by themselves.
And your effect, what you see here with shortening and not elongation of the other thing, that's also true if they're on the glass. So you get the same.
Oh,
results?
So on glass, you never get remodeling.
Oh.
For some reason, I don't actually know why. We sort of.
Well, it makes sense if they integrate, right? It doesn't.
Yeah, there's no permanent remodel. So we had to put them on this soft gooey gel for the whole tissue to actually remodel.
Otherwise, you wouldn't have gotten a result.
I don't think so. We wouldn't have gotten it.
Interesting. That's really fascinating, isn't it?
I do think it's fascinating. I mean, to me, to me, just the fact that like I always think of I never thought about the cell cycle, right? Who wants to think about that? So, but and so then you think about all the fluctuations in epithelium. You want something. This is really signaling through the cell-cell interactions that are like, that is cell cycle dependent, right? That's telling you what mechanical state you're in at any given time.
David, I'm allowed to ask another question then?
Of course.
And it's your turn to ask questions, so sorry for jumping in at the end of it. But I'm so happy to see Margaret again, and I was so happy that she was surprised to see me.
Go for it.
No, so if it's floating on the collagen, there are no integrin adhesions at all to the collagen? Is the layer, is that really floating on it? Is that?
It's not really floating. It's just the cells never spread, so that's one indicator. And if you stain for focal adhesion proteins, you just don't see many.
Okay, so you say that there's no mechanical connection.
It's not floating. It's not like at an air-water interface. You know, it's not like, you know.
But do they transfer forces to the collagen? I guess that's my question.
Oh yeah, not much. Oh, that was the other weird thing about these experiments. So like when we did this, so the way epithelial tissue is polarized, most of your actomyosin is on the apical surface, so away from the collagen. And the way we are activating, it's like a 15-year-old device. So we were doing it with a column of light throughout the apical basal plane, but the contraction was only in the apical plane and there wasn't much distortion across the apical basal surface. Yeah, so there wasn't that much. And I think that just has to do with the polarization of the epithelial cells.
Okay, well, cool. Thanks.
Great. Any other questions from person or well, I guess we so again, thank you again, Margaret.
Thank you, Margaret.
Thank you. Yeah, can you keep me on the mailing list so I can see future talks?
Yes.
Maybe I get to see Andreas talk. Okay, yeah.
You missed it already. You missed it already, Margaret.
But Andreas, we recorded yours, so she can watch yours.
Oh, no, don't show her. It's embarrassing. Beautiful, Margaret. Thanks a lot.
All right, thank you.
Maybe you should call at one point.
Yeah, thank you. How long are you in Boston until, Andreas?
I give you a call on your phone.
Okay, all right. See ya. Thank you.
Take care.
Bye.
Stay well.
Bye-bye.